Assalamu alaikum. My name is Malik Saad Qadeer and I'm going to start the presentation. Our topic is incomplete dominance, codominance, and multiple alleles. Before going to our actual topic, let's have a recap of Mendel's laws and his findings about genetical reproduction. When Mendel mixed pea plant having green and yellow color, he observed that their offspring express these two colors in fixed ratio that can also be confirmed by crossing them on Punnett scale. Although Mendel succeeded to put forward these basic concepts of genetical reproduction, he was unable to explain concepts like how a third phenotype or a mixture of these two parental phenotypes were expressed in the offspring. Incomplete dominance is one of these concepts. In incomplete dominance, the offspring has a phenotype that neither belongs to the father nor the mother. It indicates that neither of these two traits are dominant nor recessive. They blend together and make a third phenotype which is then expressed in the offspring. Pink flowers are often the result of incomplete dominance. When a homozygous red flower is crossed with the homozygous white flower, the resulting offspring will have a phenotype which is the intermediate form of white and red, that is, the pink color. Let's consider the F1 generation. To determine the phenotype of offspring resulting in the cross of two homozygous white and homozygous red flower, we solve them using a Punnett scale, which shows that all the four offspring will have a heterozygous pink.
Now let's consider the F2 generation. In order to find the phenotype of the offspring resulting in the cross of two heterozygous pink flowers, we solve them using Punnett square. The Punnett square shows that one offspring will be homozygous red, two offspring will be heterozygous pink, and the fourth offspring will be homozygous white. Hence, their genotypic and phenotypic ratios will both be 1 is to 2 is to 1. Now, let's have a look at an example of incomplete dominance in animals. When a chestnut and a white horse breed, their offspring will have a roan color. Hence, they are also called roan horses. Roan horses have a skin color that is a blended form of the phenotypes of both the parents. Incomplete dominance in humans. Incomplete dominance is a phenomenon that usually occurs in plants and animals, but it is not common in humans because human beings are genetically complex and their traits come from multiple genes. One example for incomplete dominance in humans is hair texture. If one parent has curly hair and the other has straight hair, the offspring will have wavy hair, which is an intermediate form of the two parental hair textures.
We saw that in incomplete dominance, a third phenotype is expressed in the offspring. That is a blend of the two parental phenotypes. Now let's have a look at the concept of codominance. Unlike incomplete dominance, if both the traits are equally dominant, then what will happen? The phenotypes of both alleles will be equally expressed in the offspring. Neither of alleles is completely dominant or recessive. Both are equally dominant, hence there will be no mutation of any allele, and both the phenotypes will be expressed in the next generation. For example, in camellia flowers, different colored petals are observed, which is an example of codominance. A white and a red camellia flower result in an offspring that expresses both the colors equally. Spotted horses are also an example of codominance. They have two different color patches on their bodies. These colors represent the two inherited parental alleles. Their body hair is a mixture of both these colors. Let's consider the F1 generation. To determine the phenotype of offspring resulting in the cross of two homozygous white and homozygous black chickens, we solve them using the Punnett scale method, which shows that all the four offspring will be heterozygous, black and white speckled. That is, their fetus will express both the colors. F2 generation. In order to find the phenotype of the offspring resulting in the cross of two heterozygous speckled chickens, we solve them using Punnett scale. The Punnett scale shows that one offspring will be homozygous black and two offspring will be heterozygous speckled, and the fourth offspring will be homozygous white. Hence, the genotype and phenotypic ratios will be 1:2:1.
Codominance in animals. Another example of codominance is the body color of cows. When a pure homozygous red breed cows with a pure homozygous white cows, the resulting offspring will have patches of both the colors on their body, and they are called the red and white speckled cows. Cross of a homozygous red and a homozygous white cow to determine the phenotype of the offspring resulting in the cross of two homozygous red and homozygous white cows. We can solve them using the Punnett-Square method, which shows that all the four offsprings will be heterozygous red and white speckled. This is their body will express both the colors in form of patches. Their genotypic and phenotypic ratios will be both 0:4:0, although none of the phenotypes is recessive or dominant. But when two speckled cows breed, they result in offspring that will include both the dominant black and dominant white cows. Codominance in humans. Codominance helps us understand why humans can have type AB blood but not type AO or type BO blood. In blood types, both type A and type B blood are dominant, while type O is recessive. When two heterozygous type A and type B blood types are crossed using Punnett-Square, we come to know that type O blood type is only expressed in people with homozygous recessive O blood type alleles. Also, if both A and B alleles are inherited by an individual, he will have type AB blood, for example, codominant blood type.
Incomplete dominance versus codominance. As we have seen in the previous slides, incomplete dominance and codominance have several properties that are common in both, like the Punnett square of the F1 and F2 generations and their phenotypic and genotypic ratios. Then what is the actual difference in them? Although they may seem to be closely related in terms of their properties, but they have one main difference that differentiates them. That is, in incomplete dominance, phenotypes of two alleles blend and create a third phenotype, which is then expressed in the offspring. While in codominance, the actual phenotypes of the alleles don't disappear, but are expressed equally in the offspring. After studying the incomplete dominance and codominance, we can now tell after observation that whether the phenotype of an organism is a result of incomplete dominance or codominance. So let's have a quick review and have a look at some pictures in the next slides and identify whether they show incomplete dominance or codominance. The first picture shows a roan horse. As we know that roan horses are offspring of a chestnut and white horse, hence we can tell that it is an example of incomplete dominance. The second picture shows a speckled chicken with black and white colored feathers. It indicates that both the colors are equally dominant and hence are equally expressed in the chicken. The third picture shows a flower with red and yellow petals. Hence we can say that it is an example of codominance. The fourth picture shows a pink flower. As we know that pink flowers are a result of a white and a red flower, hence we can say that it is an example of incomplete dominance. The fifth picture shows a cow with patches of two different colors on the body, that is red and white colors, hence it is an example of codominance. Multiple alleles. Many genes have two alternate forms, that is, they only have two alleles for a particular trait, but there are several other traits that have more than two alleles. If a series of the forms of a gene are situated at the same locus of a homologous chromosome, they are called multiple alleles for that gene. Although there may exist several different forms of a trait, an individual can only inherit two forms of that trait. Multiple alleles in animals. As an example of multiple alleles in animals, consider the skin color of rabbits. In rabbits, 4 different skin colors are known. Due to the existence of multiple alleles for skin color, there exists a variety of different skin colored rabbits. The agouti skin color is the wild type allele. We can see in the table shown in the slide that agouti has phenotype dark grey and its allele is represented by 2 capital C's. Chinchilla has silvery grey hair, its allele is represented by a capital C with superscripted CH. Himalayan is white colored except feet, nose and ear tail, which are black. Its allele is represented by a small c and a superscripted h. Albino is completely white and its allele is represented by a small c. Multiple alleles in humans. Blood type in humans is also an example of multiple alleles. There are three possible alleles for human blood type, that is A, B and O blood types. The human blood group is determined in the following ways. If one parent has allele A and the second parent has also allele A, then the resulting blood type will be A. If one is A and the second is O, then the result is A, which indicates that O is recessive. If one allele is A and the second is B, then the result will be AB, which is an example of codominance. If both the alleles are B, then the result will also be B. If one allele is B and the other is O, then the result will be B, because B and A are both dominant over O. In last case, if both the alleles are O, then only in this case the blood type will be O. The AB blood type is called the universal recipient because it is compatible with any blood type. It is also an example of codominant trait, as type A and type B are both expressed in it. The O blood type is called the universal donor because it can be donated to a human with any blood type. Antigens and antibodies of blood types. In human red blood cells, two types of antigens, that is A and B, are present. Depending upon the presence or absence of antigen A and B, the blood group in man is of four types, that is A, B, AB and O. We can see in the given picture that group A has B antibodies and A antigens, group B has A antibodies and B antigens, group AB has no antibodies while A and B antigens, while in group O both the antibodies A and B are present and there are no antigens. That's all. Thank you.